Okay, with few authors, there are a number of people, I won't name them all here, that have been involved with me. We've been studying salamanders for a little more than 2 decades now, some funding, and I would say that most of the people that have been in my lab, almost everybody's been in my lab, are exceptionally compassionate and are very stringent about harming anything, and we try to do this. I'd also like to dedicate this talk to my father, Isaac Blaustein. He was a Holocaust survivor, as was my wife, and when I was 6, 7 years old, he would see me running these little experiments in small jars, putting a katydid and some carpenter ants together, and it would hurt him. He'd ask me to free the katydid, and he had a very big influence on my life as far as being compassionate. And another person is from Sydney, Australia, a friend of mine, Iris Torin, who puts out at least 30 Facebook posts a day based on animal cruelty, animal rights, and vegan recipes. Okay. I probably need, I have 55 slides. I probably won't finish in time, so I'll have to go through some of these things quickly, but I would like to sort of like share some things I think really influenced my life. And one of it is a university, and in several classes we were supposed to pith frogs. You know, you take a needle and you stick it back here so you can do physiological stuff while it can't move, but it's not dead. And in this vertebrate physiology class, in a moment of panic, there was a box of about 15 frogs. I grabbed the box. I ran out of the physiology lab. I started running to this place called Passion Puddle, and the teaching assistant was chasing after me. I reached the puddle and I threw all the frogs into the puddle, and he was out of breath, and he says, "You just introduced an exotic species to New Jersey." So I screwed up a little bit there, but after that we didn't carry on with having everybody pith a frog. Okay, but the thing is that I can't read this, but maybe you can. I went through life, especially as an undergraduate, thinking maybe I'm a hypocrite because I'm reading biology books, physiology, morphology, and I'm learning things based on what other people sacrifice animals to do. So this is still something that goes through, whether I'm a hypocrite or not. This is a question of conservation. There was my ichthyology professor took us on these field trips, and he would just say, after we collected these fish, he'd just say, pickle them, pickle them, pickle them, meaning put them in formaldehyde. And then he said, we're running out of time. We're at Princeton University. He says, just dump them on the lawn. And I said, don't dump them on the lawn. Put them back in the lake. And he heard me, and he says, dump them on the lawn. So the students dumped them on the lawn. I got down on my hands and knees, throwing them in. So is this compassionate conservation? Well, not as far as the professor goes. Another thing which I struggled with is about 20 years ago, I studied salamandra, and I reached a breeding site where there was only a few liters of water, and there were really like hundreds of salamandra larvae just crowded in there. And I figured that within a few hours, maybe 24 hours, they'd be dead. So I called someone from the Nature Reserve Authority, and I asked them for permission to bring the salamandra back to my laboratory and keep them until it rained. And the person says, no, let nature take its course. But thinking about this, maybe the person is right, because is this compassionate conservation? I think that if you take such actions, you may be interfering with natural selection, for the females to choose the right habitats, for the larvae to make metamorphic decisions and metamorphose earlier. So there's some gray areas in here. So now I'll start to talk about this. In my laboratory, we've done a lot of different kinds of studies. This includes larva position habitat selection, studied how salamander larvae affects prey behavior and community structure, looked at their density-dependent effects and inter-cohort priority effects, mortality factors, life history strategies, effects of UV on salamander larvae behavior and color change, population size and dispersal, population genetics, gene expression, and the purpose of all this was both conservation-based and also conceptually based. Okay, now in my laboratory I have many people. One, Antonina is out here, and they all love salamandra. I love salamandra. I love other animals as well. And they are very, very careful about that we will not do something bad to harm them. This includes Olga, who's also in the audience here. She's been termed salamandra mom. She's always worrying about them, feeding them, things like this. Okay. So first I'll give you a little bit about the natural history of a salamandra. They're near threatened globally, they're locally endangered, and there's anthropogenic impacts that should increase a lot because the population is increasing a lot, lots of habitat fragmentation. Salamander can live to be up to 25 years old. They can be 30 centimeters, maybe a little bit more. It's active on rainy nights, and the females are resting someplace during the summer. They come out when the rains come with a complement full of babies. They migrate to the breeding sites. The males arrive first. There's probably some evolutionary reasons for that, but maybe a physical reason is the mama's real fat, so it's hard for her to get there. They breed in many different kinds of sites: temporary pools, permanent pools, running water, stagnant water. But they don't lay eggs, they larviposit, and they can larviposit up to as many as 200 tadpoles, larvae, babies, whatever you want to call them. Then they make love, and then they leave the breeding site. The larvae stay for at least 10 weeks in the breeding site. They have to metamorphose before the pool dries, or they'll die. But in permanent water, a fraction will stay throughout the summer, and they can metamorphose in September, October, when the first rains come. We know very, very little about the post-metamorphic, the juveniles. It takes about 3 to 5 years before they become reproductively mature, and we really know almost nothing about them. Okay, so again, I want to focus on the methodologies. So we are interested in estimating population size and also looking at dispersal. The conventional wisdom was there's very strong breeding site fidelity, so that all the populations were, what's the word? In closed systems. But actually this is not the case. But for marking them, we didn't have to cut off any toes. We didn't have to do things like this. That each salamandra has its individual specific spot patterns. So we would take photographs, and we would be able to see later whether we had the same individual, number 28, or a new individual, or whatever. And using this, we're able to estimate populations. This is work done by Iftach Sinai. He showed that permanent breeding sites support much higher adult populations than temporary sites or semi-permanent sites. We also use the spot patterns to look for dispersal. This work was done largely by Shirley at the beginning, and essentially what we were interested in is how far do they move? Are they subpopulations? Are there metapopulations here, or are these closed populations? Initially, this was some of the work that was done on Mount Carmel, and we found that I'm sure that as we do more, we'll find more, but about the maximum distance that they travel is about 1.3 kilometers, and the breeding sites do a radius of 1.3 kilometers from the center. You can see that many of these populations there's interbreeding going on. So also we also found that if we wanted to do work on the salamandra larvae, which is what I originally started doing, is that if you wanted to know each individual, you create these mesocosms, you want to know which is each individual, that every individual has specific tail spot patterns. So we're able to photograph them and later determine which individual they are. So we do things like we studied mixed cohorts to look at density-dependent effects, priority effects. Again, I won't concentrate very much on the research. Ori Segev did studies to try to see in intermittent wadis, will they disperse downstream and find other breeding sites? And they do to some extent. Some of them stay, some of them go missing, etc. But again, what we did use is the tail spot pattern in order to be able to identify every individual. Okay, this is a story that I am half comfortable telling. It's about the effects of gambusia on salamandra larvae. So initially, some people, legally or illegally, they put gambusia in some salamander breeding sites. And what I did it in 1999 and 2003, and found that there were 0 in some, and there was a lot of gambusia in others. And what I saw was that I don't know if you can see it very well, but the gambusia take nips from their gills, from their toes, and mostly from their tail. So we look at tail-body ratio. We see that where's my cursor? Never mind. That where there was lots of gambusia, the tail-to-body ratio was a lot smaller because they're eating the tail. And then I did this study before the gambusia was introduced in 1999, and after it was introduced in a particular pool, they basically, the survival is 0 when there's high densities of gambusia. None of the larvae make it. So I said to Ori Segev, this is not really an experiment, but we have some rather strong evidence here. Let's send it in conservation. Anyway, there's info gap theory. You don't always know everything, but you need to act. So we submitted to the Israel Journal of Zoology, and we presented the evidence that gambusia was bad for salamandra, that salamandra anyway ate mosquitoes, and that gambusia should be removed from any salamander breeding site, particularly because they're endangered species. Well, the paper was rejected, and the reviewer said, you need to do an experiment. So I said to Ori Segev, okay, let's do an experiment. So the experiment was, we took gravid females, we let them reproduce in the laboratory, and the treatments were no gambusia or gambusia crossed with the presence or absence of artificial vegetation. Okay? These were in large mesocosms, and what we found the same thing is that the gambusia love to eat the salamandra, just nip at them. The survival rate, regardless of whether or not there was artificial vegetation or not, was quite low compared to the absence of gambusia, and of course the tail-body ratios, etc. Survival was really, really bad, and we submitted this paper then to a biological conservation journal. But the editors of the journal said they wouldn't consider it because we were causing harm to individuals of an endangered species. So we had some correspondence. Okay, I can at least tell this story. I wrote back several times trying to explain to them, say like, I thought we were the good guys here because in the field itself there was 100% mortality, but in our experiment there's only about 50% mortality. So in the end, we saved many of the salamandra by actually doing this experiment. He didn't understand me. He said, I told him we submitted it somewhere else, but I just wanted to understand. He said, and then I spelled his name wrong. He got very angry, so that was the end of that. And we published it in the Animal Conservation. So so I came up with a formula, which you people can tell me that I'm wrong. So the formula is, if an experiment sacrifices fewer or equal number of individuals that would have occurred in nature anyway, and the information accrued from the experiment that sacrificed some individuals leads to better conservation management and fewer mortalities in the future, then I would call this compassionate conservation. Okay, you can answer that later. All right, we have done stuff with roadkills. This is a lot of work by Iftach Sinai. We want to get these underground tunnels. So far, it hasn't happened. We checked various. My time's up, right? So we took just a little shtickle, a little piece of the tail for microsatellite data to look at conservation genetics. We've found big differences. We're continuing doing this. And then an interesting study was that we now have we're finishing up a grant on ecological transcriptomics, ecological genomics, and we were interested in studying gene expression. The influence of gene expression on risk of predation, desiccation, dissolved oxygen, many things. And our German collaborator said, in order to do the gene expression, you have to sacrifice the entire animal. And I said, we cannot sacrifice the entire animal. The Nature and Parks Authority won't let us do it. Isn't that right, Noam? Yes. Thank you. So I insisted on just taking a small tail fin clip, and they said it wouldn't work, but it did work. If you look in the center here, many of the genes that were expressed in the tail, and for the last few years, we've done our gene expression studies using just a tiny bit of tail fin. And Ori Segev showed that cutting off a little tiny piece, the salamandra are cannibalistic, so they bite each other's tail anyway, that the mortality and the size of metamorphosis and everything else doesn't change. So in summary, we use a number of methodologies which I hope is good, and I think that we do ecological studies which will be good conceptually and also good for conservation. Yeah, okay, yeah. And love salamandra, and thank you.